Okay, so welcome to our first concurrent section, Genomics, Research, and Computational Challenges. This is a 90-minute session. Our speakers for this section will be Cavalcoli, Gonzalo Abecasis, and Andres Wierzbicki. And I'll turn it over.
Thank you very much, and thanks to the organizers for putting this all together. It's been a wonderful day so far, and hopefully this session will be equally enjoyable. So one of the things that we kind of tossed a coin at the beginning to see who would go first, but in a sense, my talk is a very high-level overview that sort of sits at the bridge of a lot of the computational things that we've heard earlier this morning, as well as the genomics and biomedical side of things. And so I'm going to touch on a variety of things today and hopefully give you some food for thought towards the end. And I'm not going to go into a lot of experimental details or data today because this really is meant as a high-level overview of where does computation and genomics and medicine kind of intersect. As a bit of background, I'm the director of the Bioinformatics Core and a research faculty here in CCMB, our Center for Computational Medicine and Bioinformatics, and soon to be department. So overall today in this 25 minutes or so that I'm going to talk, I'm going to cover genomics and the technologies that are used for analysis and a fairly small subset of genomics. And I want to talk a little bit about setting the perspective of the past, present, future, because in essence, the things have changed a lot in the last few years, and I don't think we're done changing. So, and then I want to talk about the role of computation and analysis. So I'll talk to you both about the analysis pipeline in a sense and how computation intersects that at different points. And then I really want to spend a fair bit of time talking about the challenges that we're facing, because putting the challenges out there for people to think about and ponder about how do we address these challenges is one of the great benefits of having these CI days, is putting that information out there and letting multiple minds and good minds think about solutions. So first of all, to set the stage in a basic sense, genomics refers to the measurements of either RNA, expressed RNA in cells, in tissues, genomic variations at the level of genomes, looking within the genes themselves, or in many ways in environmental and other microbiological samples, whole genome sequencing, either de novo or with reference genomes. And so I'm going to use genomics in that loose sense. Many people would fine-tune that a little bit, but for today, give me the benefit of the doubt, and we'll look at this as a large picture. There have been a lot of trends over the last 10 years or so that have radically changed the amount and the cost of DNA sequencing. And it's hard to pin down the exact point, but I'm going to give an approximate 10-year time frame for this. And so I want to set the stage, given the nature of the season and the fact that we were dealing with past, present, and future, I want to bring you to a picture and a trend I'll have along here. First off, though, because I didn't know the exact nature of the audience today, I'm going to talk a little bit about terminology. As computer scientists and engineers, we oftentimes talk of mega, giga, gigawatts, gigahertz, gigabits, but we actually can get confused because we all get into our own jargon. And so I prepared this slide when I gave a lecture in a class a year or so ago, but it makes sense just to have in there. I won't dive into too much detail on it. But when somebody says, if they're in the DNA sequencing core, and they said, yeah, I just generated, 100 gigs, they don't mean 100 gigabytes or 100 gigabits or anything like that. They mean 100 gigabases. And so we tend to put these prefixes in front of things and develop our own jargon. And so be aware that people can toss out the same terms and mean different things. Many people refer to, and this gets down into the biochemistry level of things, people will call something a nucleotide or call it a base or call it a base pair, and in general they'll mean the same thing. But as I was teaching to students in graduate school, I wanted to emphasize the fact that there are differences between them. We shouldn't lose sight of these differences and let the jargon degenerate. So make sure you're clear about what you're saying when you say it. And I won't go into any further detail on that unless somebody wants the biochemistry lecture, and then we can do that at the end. So in the ghost of Christmas past, we think, and it's not that far ago, this doesn't reach 100 years into the past. Sanger sequencing was first published in, I believe, 1977, so that isn't that long ago. And as recent as the Human Genome Project, most of the sequencing was done in fluorescent capillary well sequencers that could do 384 clones one in each well per run, and they could run many of these runs in a day, and yield about 700 to 900 base pairs of bases of DNA. and they were read by fluorescent image. And so the total output from perhaps a week of sequencing might have been 2 or 3 gigabases of DNA. And the output of that would have looked like these fluorescent grams, and you would call the bases depending on what color it was and where a peak was detected. So the present, and in some ways the ghost of present, is that we're in a position in technology where we have massive parallel sequencing of shorter fragments. So we're not going to see these large 700 and 900 base pair pieces, but we've made several advantages and changes in the technology. Number one, the fact that you don't need to subclone the DNA to start with. Now, for those of you that haven't been in the lab and used pipetters, what we mean by this is that in the past we had to take DNA, break it into pieces, and clone them into a bacterial vector, grow them up in bacteria in little cultural tubes, isolate the DNA, and then sequence it. So the number of steps and the amount of time it took and the amount of robotics and reagents was quite high. Now we can take this DNA directly, add linkers to the end, amplify it, and sequence it. The cost per megabase of sequence has gone down dramatically. I'll show you a graph that is almost hard to believe, but it is true. And the amount of megabases of DNA per day that can be generated with this technology has increased as well. I will say as a caveat, there's been a slight increase in the error rate per base, but the amount of data that is generated is so high that those that don't match can be filtered out and discarded without a high loss of, or without a large loss. There are many of these platforms available. I'm not going to I'll show you some of them today, but I'm not going to discuss any of the specific technology platforms. Just to give you a sense of how much data we're generating, in the past I said that a Sanger sequencing run might generate 2 gigabases or 3 gigabases of data in a week. A HiSeq run, as I'm going to show you here, this HiSeq run, HiSeq 2000, one lane, and there are 8 lanes of it, so multiply this by 8, generates about 1.5 1.5 times 10 to the 8th reads per lane, multiplying that times 8. So the amount of data we're generating is almost on the order of 100 times larger than what we were getting out just 5 or 6 years ago. If each of these is only 100 nucleotides long, we can actually generate 3 times 10 to the 10th nucleotides in a single lane of data, multiplying that by 8. So we're upwards of almost hundreds of gigabases of data that are generated. A run takes about a week, so you have to factor that time in, but still the amount of data being generated is significantly higher than the past. Given this and the coverage calculation on this slide, the size of the human genome is large at 3 times 10 to the 9th nucleotides, but as you can see with some of the numbers we're generating, we're actually getting upwards of almost a genome size of nucleotides per run. And if we're only looking at the RNA or the exomes, we can easily look at somewhere around 500x coverage per lane of sequence. So the numbers vary widely from the labs, but that's the general order of magnitude is the amount of sequence being generated. While we cannot sequence a single individual in one flow cell yet, we're probably getting closer, and I'm sure Gonzalo will talk a little bit more about some of the throughput from the 1,000 Genomes project. Here's the slide that's a little hard to believe, and as we heard this morning, and we saw the actual notes from Gordon Moore's lab notebook, this is a line which draws in some ways Moore's law, which is it basically deals with the decreasing cost of production per transistor. In this case, it's the cost of sequencing per megabase of DNA sequence, and this comes from the Human Genome Research Center. And in this case, you can see that if we look back only as far as 2001, this does not go back to prehistoric times, but we were dealing with just under $10,000 per million base pairs being sequenced. And then somewhere in 2007, as next generation sequencing really hit the heyday, the amount of the cost per megabase, you can see precipitously drop. The slope changes dramatically here. I'm not sure it's quite down to 0 yet. I believe this is a slight artifact of the graph, but the slope has stayed the same, and so we're down probably almost a 1,000-fold decrease in the cost per base in just the last 4 or 5 years. So I don't know that that slope was going to get any deeper. Otherwise, it's going to go the other direction. And
actually, they know. Third gen hits, it will change again.
Yeah, and I'll talk a little bit about my ghost of the Christmas future. Thank you for that segue. So this is the ghost of the Christmas yet to come. And in the future, one of the things that's out there right now, and it actually is here, is single molecule sequencing. And currently, that we have a Pacific Biosciences or PacBio machine here on campus, and we're beginning to pilot data coming off of that. There are some sequences available. Right now, the current error rate associated with the long reads makes it not useful for really, really long reads. You have to actually circularize the reads and do a lot more. I won't go into that technology. But there needs to be some improvements in these single molecule sequencers. In the past, what has happened was, and to get into the technology a little bit, in the massively parallel sequences, you put millions of isolated clonal forests of the same DNA molecule on a glass slide, and then the polymerase is in the solution around that. In these single molecule sequencers, and I don't have a diagram of it in my slides, the polymerase itself is actually is anchored to the surface of the flow cell, if you will, or the surface, and the DNA passes through that. And so we're beginning to see changes in the technology. Some of the future ones include, there's a company called, a technology called nanopore. In this case, the DNA is actually passed through a pore in a membrane, and the changes in ion potential are measured. Visagen is another one that is dealing with a slightly different technology, and there are some coming out that deal with sequencing by massive hybridization to probes, mass spectrometry, even electron microscopy or atomic force microscopy. These are ones that aren't even named as technologies yet, but people are investing in them. And fairly recently, the Archon X Prize was announced, and the contest will award you $10 million if you can come up with the following. You have to sequence 100 human genomes within 10 days or less, with an accuracy of less than 1 error in 100,000 bases, covering 98% of the genome, and at the cost of less than $10,000 per genome, which I think all of those together, there's a lot of tens and hundreds in there, but that's a pretty high challenge given today's current technology. What are you thinking, Gonzalo? Think you can do it? Okay. All right. Here's a slide that I have to change every 6 months. And that is actually one of the problems being in this business, is that I can't use the same slide deck from year to year. People catch me on it all the time. That technology's gone. This one has changed. Where's the new one? And so every time you give lectures and talk, you have to actually change your slides. And so I've had to take out technologies and add ones in. In fact, I believe, I don't think there's a Helicos machine out there right now. I don't know quite where that company is at anymore. Pacific Biosciences. Life Technologies, Ion Torrent, as well as the Applied Biosystems. So there's a lot of different platforms. Each of them have their pluses and minuses. My talk is much too short to go into all of that, but it is out there to know what the different technologies are for the choice. But all of them are generating a larger amount of sequence and have different pluses and minuses. But all that technology has to be applied to biology in some way. So genome resequencing is probably the largest amount of stuff that comes into our bioinformatics core. RNA expression, ChIP-seq, protein binding to DNA. These are things that are out there, and then the technology is being applied with high throughput sequencing. So what is the role for computation in genomics? And I think my number one right now, my day-to-day challenge is storage. And that is the fact, how are we keeping this data and where are we keeping this data? A single run, like I was referring to, that one lane that generates gigabases of data, a single run from Illumina with all 8 lanes generates almost 1 terabyte of data a week. And you have to hang on to that data and do something with it. When you actually process the data, you increase the size of the output files. And so a lot of times now, so storage is a challenge. I'm not sure that's an insurmountable one. It's just, it remains a challenge. Data transfer. I refer to SneakerNet and FedExNet. Because while computers are great and networks are great, if you're transferring 2 or 3 terabytes of data, it's still faster for you to put it on a removable drive and walk it over to my office. Or if you're going to put it across the country and you've got 10 or 100 terabytes of data to transfer, it makes more sense to actually move the drives than it does to ship the bits across FTP or across internets at this point. Those are 2 challenges in the initial data, but once we actually get the data in hand, aligning data to reference genomes, assembling the reads, whether you do it to a reference genome or de novo assembly, calling peaks, calling the transcripts, and right now visualization and the downstream analysis, once you get the peaks called, determining the sequence variants, copy number variations, structural variations, all of that remains to be computationally. As of now, in the software that currently exists out there, the aligning reads to reference genome is a parallelizable a process. Assembly of reads de novo may possibly be parallel ready, but it's not quite now. Most of it requires these large de Bruijn graphs and graphical structures requiring lots of memory. So there's still a role for computation, but there isn't one clear pipeline, one clear cluster analysis. I'm going to skip through this. I will talk about this as we go, and I'm running a little short on time. The data itself, when you get the data out, actually looks like this complicated slide. This is a FASTQ format where the nucleotides that have been read by the sequencer are here along with quality scores. I won't go into how the quality scores are presented here, but they're the probability that the base is called correctly. And it's in what's called the Fred notation from the old human genome days of Phil Green's editor, and they're in the same vein of the probability of error model. So because we have quality associated with these, we run some QC on these reads as we get them. And given the length of the sequence read from left to right, and the box plot here represents the error that we see, the mean error and the related error associated with all of the reads in the entire run, so we get a sense of overall quality. The aligning and assembling of reads to a reference genome is really the next major step that we do computationally. If we've got a de novo organism or something where we don't have the genome, or we're looking for something very novel, we can do de novo assembly not using a reference genome, but actually using the reads themselves to create the assembly. And there are numerous different tools available for this mapping and aligning. We primarily use either Bowtie or BWA for our aligner, for reference-based aligning, and for de novo assembly, Velvet, Abyss, etc. Really? Okay. Okay, I got 9 minutes left and you got 1, so we're going to average. I'll go just a little bit over and go quickly. Sorry. I'll skip how we reference them. There is standard formats for reporting the alignments of reads, the SAM, sequence alignment and mapping format. The links are in my slides. There are a lot of tools that we use for the downstream analysis. After we align the reads to the genome, that is relatively easy these days. It's computationally somewhat intensive, but it's very doable. But each experiment that comes in requires a different analysis, and there are a lot of different tools for the downstream analysis of genomes. And visualizing the reads, while a lot of the analysis is still done in tabular form and a command line, a lot of times you want to look at the reads and see and verify for yourself. And so there's a few tools that are available that are also listed in my slides as well. Okay, I want to get into the challenges though. So there are 3 major challenges I want to point out today. The first I've already alluded to, and that is the data issues, storage, the access to the data, transfer of data, life cycle, how long do we keep it, and eventually privacy and risk associated with data. How do we translate what we're doing at the research level into medicine and applied medicine? And the second is the accessibility and usability of this data and the computational tools. And that's really a main one for this audience. There's another graph that is somewhat scarier than even the first one I drew about the cost, and that's at the amount of data being generated. And this graph shows, this is taken out of Lincoln Stein's paper from about a year ago, but it basically shows that in the olden days of Sanger sequencing and pre-next-gen sequencing, the slope of the increase in the amount of data was actually below the slope of the cost reduction in the amount of storage. So it actually cost you less next year to store it because the price of storage disks went down. Somewhere around 2004, 2005, the inflection point changed with next-generation sequencing, and the rate at which we're generating data is now exceeding the cost reduction in storage, and so it will cost you more next year to store the same amount of data because, it'll cost you more next year because you'll have more data and the disk prices aren't going down fast enough. Okay, privacy. This one I really want to get to, and I may skip through some of the others, but while DNA itself does not identify a person, if the pattern in DNA can be detected and compared with another pattern, you can actually say that this is the equivalent of a fingerprint. And so the point at which we can link 2 samples together and say they are identical can be considered an identifier. And there's an ongoing national discussion about whether the DNA sequence information will become one of the HIPAA identifiers, which will then cause us to have to deal with the data in a different fashion in order to comply with regulatory guidelines. And so that's just an awareness piece that's out there. It isn't law yet, and it's still under discussion, but it may change how we have to handle data and what we have to do with it. Translating results to medicine. Linking genomic data to other health information. How do we integrate genomic data with other patient data, disease state, progress, efficacy of treatment, phenotypic measures, and that leads us directly to personalized genomics. How do we actually take this data and look at individual people and their family structures, their susceptibility or their susceptibility to disease or their possible treatment route? And then I think this is still on the horizon, but not that far on the horizon. It's kind of like snow. It sort of shows up right before you expect it to. But genomic data is part of the electronic health record. Will we actually carry around with us on our iPhone someplace an application that contains portions of or all of your DNA sequence as part of your health record? And how will we deal with that information and data? Finally, yes, there is one tomorrow on that as well. Finally, I think for the bringing communities together, biologists, and I won't insult the biologists in here, but biologists on average are behind the curve on learning, adopting, and using high-performance computing, especially clusters, schedulers, PBS scripting. If you talk about PBS scripts in front of a biologist, they say, wait, is that phosphate buffered saline? Or, in other words, the acronyms collide again. But on the other front, I think we have to bring the groups together because not every problem can be thrown at a cluster. And so we have to bring together the communities and come up with ways, and I think that in a lot of ways what we have to deal with are my two subpoints here, which is there is a learning curve and an onboarding process of getting biologists to use computers to better their advantage, and the second is we need to develop more diverse, flexible, and adaptable resources for storage, networking, and processing of data. And I think those are the two biggest challenges. And in one way to address those and where we're proceeding with this is that there have been some changes in the last year or so with a new MSIS structure and the new med school CIO, Ted Hans. There's been investments in both personnel and computing resources, including Jeremy's in the audience here, a high-performance computing specialist for the medical school, helping to bring people to Flux and help bringing Flux to us. There's a new Department of Computational Medicine and Bioinformatics, and there will be a new Bioinformatics core, both of which will be kicking off in January. And so those are kind of shameless plugs, but at the same time, understanding that there is a commitment here at the university for doing this, and there are mechanisms for bringing more and more of researchers together with this computational infrastructure. And I think for the sake of time, I will stop there and take any questions we have. All right? Thanks. Thanks. Quick questions before we move on?
Jim, what do you see as your biggest challenge going forward in the next couple of years, other than cloning you?
I think it's going to be dealing with, we can align sequence reads to genomes. We can do a lot of that. There's a couple areas where I think we're going to see big need for change, one of which is in metagenomics and sequencing of community structures, because that remains kind of a black box of understanding. How do we understand what's going on with the whole community of organisms? And secondly, I think the diversity of downstream analysis, what we've done is move the bottleneck forward. We now can sequence the heck out of things and get lots of data. We still have to analyze it and then apply it to biology. And so I think there still remains a large black box of biology. And I think if I were standing on a soapbox, which this is kind of a large soapbox and I get a chance to do this, is to say that I'm cautiously optimistic that high throughput genomics can actually help us address the black box of biology in terms of what proteins interact with what proteins, what's in a pathway, what is disease causing, what are the relationships and interdependencies between disease and biology by actually tapping into some of the high throughput data. So I think that's a challenge as well as a possible bright light. Yeah.
What do you think about, on your third point there, the platform, say Galaxy for data management and addressing that need? I guess, what further is needed beyond something like Galaxy? I guess is kind of my question for what you're talking about here. What further is needed?
Galaxy, I think, is a good starting point for a set of tools wrapped up. I think it covers a lot of the very basic things that would be done. I think one of the concerns I have is where is the data being stored and for how long and who has access to it. As we go forward, we're going to have to look at the data management side of things and the data lifecycle side. And Galaxy doesn't address that really well. You still have to upload files and move things back and forth, either in and out of a public server or your own server. And then the question is, how long can you leave it there? The compute cycle actually is less of a demand on things these days, although you still need a lot of it. I don't think we're running out of compute cycle. I think we're data movement, data storage is a bigger issue. But Galaxy represents a good starting point. I think right now the only problem I have with Galaxy is that it has a fairly defined set of tools, and when I want something outside of that, I have to then take everything out of Galaxy, go someplace else, and bring it back in. Or I have to spend the time to write and wrap what I want and put it in Galaxy, and I oftentimes don't have that time. But that's, I think a set of tools like that might be beneficial. GCG was used for a long time, and it became a very de facto set of tools, and Galaxy could easily become that. So, Lee?
Given that the SRA is basically being phased out and it's kind of on life support right now, what do you think the future is for data
So I wish I had the NSF guys and some NIH guys videoed in to ask that direct question. Long-term storage and public repository that extends beyond the grant life cycle is an issue in public funding right now. I don't know. I don't know what's going to happen with the short read archive and the public repository for data. It's an enormous amount of data to try and hold, and unless you have some sort of policy towards it, I think we won't see it today, but at some point you're going to see some kind of compression strategy where we don't store all the genome, we store what's different, or something like that, that something's going to come forward. Either that or somebody's going to invent the Star Trek Ridgeline crystals and we can store a lot more data in a cube. One of those things. Chris.
I've actually heard that UCSC is building a genome hub and they will be transitioning SRA over into the genome hub. I don't know where that's at.
Okay.
But along those lines, one of the things that UCSC is doing is providing an analysis environment, basically a cloud environment. And that, from my perspective, as a secondary consumer of data, not someone who's generating these sequences, but trying to analyze them. One of my biggest problems is actually the movement of data. I spend enormous amounts of compute cycles just moving data from one place to another to do a trivial analysis that produces a very small bolus of data. So do you see a trend towards not data transfer, but analysis transfer, where you're moving the analysis to where the data lives, not worrying about that?
You know, Chris, this came up as, this actually goes back a couple of years. I asked a similar question. There was a high-performance computing thing at Harvard, and a lot of big brains get together about this, and I said, you know, what are we going to do about moving this data faster, and can't we widen the pipe to send the data over there? And the guy just was kind of like rapping me on the forehead going, just move the analysis to the data. And so this is where I think that we have to be smarter about how we do the analysis. And I'm not against cloud, or I don't even want to take a stand on it, quite honestly. But I think that we have to address what are the bottleneck issues. And data movement of terabytes of data isn't going to get substantially faster. I mean, you can bond a couple of different FTPs. There's Globus FTP. There's things we can do that will incrementally, linearly speed it up. But when you scale, it still takes a period of time to do that. And is that worth doing relative to moving the analysis closer? And I don't have a solution, but I think that is a challenge. We have to address that because for doing one experiment, it's okay to move it and do one. But if you've got to do these things in a pipeline and repetitiously, you have to move the analysis close to where the data sits. You know, in our bioinformatics core, we have a storage array that sits right snuggled up next to the analysis machine, and I move the data once. I don't even move it into project directories. I create soft links and symbolic links. The data moves once because that's a cost. And I don't duplicate the data because it's a cost. And you know, if we get into issues of if Amazon becomes capable of taking on HIPAA responsibility and people are willing to move their data there and take on that risk, that's one possibility. But I personally like to mitigate risk by keeping things relatively close to me. I think that's more definable, but these remain possibilities for solutions, okay? All right. Thank you very much. Salo is next. And just tap the screen. Yep.
All right. So I was asked to talk a little bit about how we're using next generation sequencing to study complex traits. I think it's a varied audience, so I tried to first just give a high-level view. You know, what are the analysis steps that happen to the data early on, give you a sense of the computational cost for the data sets we're dealing with, and then walk through a couple of examples of specific analysis. You know, just to say, you know, like you heard from Jim, the scale of these data sets is just staggering. You know, so the first human genome was sequenced ending in about 2001, 2003. Took, you know, 5 years or something like that to do. We think that over the next year, we're going to have sequence data on something like 20,000 genomes to analyze here. You know, so the scale for that from that 1 to 20,000 is pretty staggering. Okay, so as you heard, we have very high throughput sequencing technologies now. There's a number of commercial platforms that allow you to do this. The thing that they typically have in common is that the reads are relatively short. You get millions of them, and the error rates compared to traditional sequencing are relatively high, maybe on the order of 0.5% to 1%. If you're studying human variation, if I compare DNA from any two individuals in this room, I expect to see a difference about every 1,000 bases. So an error rate of 1% means that I have 10 times more errors than real signal to start. So I get this pile of reads, and the first thing I need to do is to somehow align them relative to each other. Each read on its own has very little information. So the first step in these analyses is basically read alignment. I take each read, I try and place it somewhere in the genome, find the best matching placement. It's a relatively straightforward problem with good solutions. You might imagine that if you took tens of millions or hundreds of millions of reads and did the scan across the genome, it would be a terrible solution. But once you build an index of words and say, for each 15-letter word or something, I'm going to list all the positions where it occurs, you can take your read, look it up in the index, and place it relatively quickly. So there are some nuances to this, because it turns out that placing each read on its own often doesn't give you a very good solution. And so after you do the initial placement, you typically look at groups of reads that align over the same location and try and refine the alignment. So here's an example. If you look at these bases here that are highlighted, they're highlighted because they would mismatch the reference. And there's a decision to be made, which is maybe instead of putting a mismatch, I can insert a little gap and remove the mismatch. Now, if I look at one read at a time, I'm going to say, well, actually, mismatches are more common than gaps. And so that would be the right decision. Once I see a pile of reads and I have other evidence that there is a gap there, I can resolve this, which would have been hard or impossible to do otherwise because it's at the edge of the read, and say that's probably a gap. So once we place them, we usually scan over them and try and refine the alignment in various ways. There's another kind of refinement that's similar, but that doesn't rely on having deep enough coverage of a location. And this is, when you place a read, you can say something about how confident you are of that placement. And that's got things to do with, well, how much better is the placement that I chose compared to the next best placement, or the next best placements if you have many choices. It turns out that you can actually have uncertainty at the per base level. So it might be that you are very confident that the read maps there, but you're less confident about what happens to individual bases in that read. So let's focus on this little g here at the end. It's been aligned to a C. That's one possibility. Another possibility is to place a gap, just like before. And I have to decide between these two. If I have just one read, I'm going to make a choice, but I'm not going to have very much confidence in it. And so one way to do it, and here in this cartoon, I don't know if you can tell, but the G is shrunk a bit, is to say, I'm uncertain about that particular detail of the alignment, so I'm going to reduce the quality for that base, and I'm going to discount it when I'm using it to discover DNA variation and so on. And this actually makes a huge amount of difference. When we started doing things like this in a 1,000 genome project, we removed about half of the errors that we were making in calling variants. Okay, so assuming you do this, you can place the reads, you can refine the alignment, you can calculate this per base alignment qualities by summing over all possible alignments for each read and so on. The last thing you want to do is to call variants. You want to identify positions that vary and make judgments about those. So, in theory, you might say, hey, I'll have a pile of reads at a particular location. Here in the position marked with a star, some of the reads say there's an A, some of them say there's a C, and I want to make a judgment about the individual that I extracted the DNA sample from, and that was used as a template for those reads. And the judgment might be maybe that the individual is a heterozygous, that he has a chromosome that's A and one that's C. But to formally do this, I'm going to build a probability model. I'm going to take the reads, calculate their probability given each possible true genotype. Before I have any data, I say, hey, any set of reads is equally likely, or they're equally likely given any possible true genotype. And as I accumulate, I say, hey, the first one says a C. What's the probability of that if the truth was homozygous for A, or if the truth was homozygous for C? With an error rate of 1%, those probabilities might look like that. And I gradually pile in the reads and update those probabilities. At this stage, I've got probability of the read set given each of these 3 genotypes, and I'm almost ready to make a decision about what to say about this base. And what I need to do is to combine it with some kind of prior information. So I'm going to say something like this. Before I have any data, I expect that most positions would match the reference, because the reference is just a chromosome from a particular individual, and I know that any 2 individuals differ very rarely. So I'd say, without knowing anything, that I expect 999 times out of 1,000, the individual will match the reference for both alleles. And only 1% of the time would there be a mismatch, but most of the mismatches would apply to a single chromosome. So if I combine the probabilities I just calculated with these priors, actually I'd say, hey, with very high confidence, 80% probability, this individual is not a heterozygote. He's actually homozygous for the reference. Because of this very strong prior for matching the reference, to discover variants, one needs quite a bit of redundancy. In fact, to be confident of finding most variant sites, you'd probably need to sequence a single genome about 30x redundancy. And so your 3 gigs of data become 100 gigs of raw sequence for calling one genome. There's different ways to actually interpret this. And so a different way to take the exact same raw data, but to come up with a different conclusion is to say, hey, why don't I look at what happens at this position in other individuals? If it turns out that the position varies, and particularly if the position varies in individuals who have a similar flanking stretch of chromosome, I might have a slightly different prior. I might say, I already know that this position varies in, say, in this case, 20% of chromosomes. So I change my priors, and based on changing my priors, I can now actually make a confident, different decision. So it's the same data interpreted in different ways. Okay. So when it comes to studying a genome, these are the set of choices that you now have. So you can do deep whole genome sequencing. If you just take the data generation cost, if you are savvy about bargaining with the different providers, right now this costs about $2,000 a genome. It's the most complete analysis, about 30x coverage for the entire genome or something like that. You could say, hey, instead of trying to do this over the entire genome, because $2,000 still adds up. If I want to do this over 20,000 individuals, it'll be $40 million. If anyone here in the room has those $40 million, I'd be happy to take them. But if not, I can try. Anyway, so you could say, hey, why don't we focus instead of on a subset of the genome that's particularly interesting? So you could do exome sequencing as an alternative for quite a bit lower cost. Or you can say, well, I'm actually going to do the entire genome, but I'm going to do it at lower coverage. And I'm only going to be able to interpret it by looking for these shared stretches and so on. It turns out that now that there's enough sequenced individuals out there, you can actually build new kinds of genotyping arrays that look not just at common variants, but also at rare ones. And those are also kind of an interesting take on this. OK, so just looking at what we've described so far, I'm going to try and give you a sense of what we use for our computing needs day to day. So we've got a compute cluster that's shared between my group and another research group in our department. We have about 2.5 petabytes of storage, 5 terabytes of RAM, and about 1,500 CPU cores. It's been customized quite heavily to support applications that do a huge amount of I/O. And we use Flux for overflow. So I think right now the maximum you can request from Flux is 500 nodes, and many months we request that. Most other months we request about 250.
We're going to increase that.
Good. We'll use it. So if you say, what does a project, say, with 1,000 samples currently take for analysis? So when we take, say, the current set of 1,000 samples sequenced by the 1,000 Genome Project, that corresponds to about 60 terabases of raw sequence data. If we transfer it in here from NCBI, for example, it takes about 2 weeks for data transfer with about 4 terabytes a day or something, so we can get. If you say, well, the initial placement of the reads, finding their best alignment, refining the alignment, and so on, takes about 300 CPU months over these 1,000 samples. Calling small variants takes another 200 CPU months, and calling larger ones takes about 300 CPU months. So I'm measuring this not in hours or minutes, but in months. It kind of works well. And then the final step where you kind of try and refine genotypes at each individual by looking for shared haplotypes and so on takes 400 CPU months. So if you had about 1,000 dedicated compute cores, you need about 6 full weeks of computing time to do this. And so that's one project, 1,000 samples. If you say, hey, what about if I want to do 20,000 samples over many different projects, it gets to be a bottleneck, and you see that, hey, it'd be good to think about ways to do this even more efficiently. And of course, you can get a lot of computing power going back to the Amazon cloud or something like that, but the costs are actually also rather high. So I think we estimate it costs us about between $50,000 and $100,000 for this 6 weeks of compute power on the cloud. Okay, so here's one of the projects where we're involved in, the 1,000 Genomes project. Basically, the goal here is to build a catalog of all genetic variants, and this is kind of an essential resource for disease studies. We published our initial report at the end of last year in Nature. We, at the time, had sequenced 2 small families. We call them trios in the genetics lingo, basically parents and offspring, and another 180 genomes. We discovered something like 16 million genetic variants. Some of these were single base, 15 million of them. Some of them were multi-base indels. If you go look right now, even though this paper was published in November, it's the highest cited paper in Nature for last year by a couple hundred or something like that. How much do we gain by doing these analyses? So if you looked at the data with a standard analysis that basically looks at a site at a time, tries to adjudicate what the genotype is, this panel here gives you the error rates. So the error rates depend on frequency a little bit. You can see that at heterozygous sites, for example, we'd get the right answer about 80% of the time. Once we refine the calls with those last 400 CPU months of compute power, we reduce that error rate from 20% to about 2%. So that's nice. There's a couple of other interesting things when you look at this type of data that are kind of fun to think about. So when we started the project, we had different groups who had their own analysis tools, and we were sort of set out this strategy where we said, we'll evaluate them, pick the best, that'll be our official answer for the project. So there was a team at the Broads, one here in Michigan, one at Sanger. And so we each analyzed the entire data set and made a set of calls. And then we went out to generate a truth set for a subset of those and evaluate. What you can see is that depending on what metric you choose, you end up with slightly different answers. So if you look at homozygous reference errors, well, the Broad here has the lowest rate by a hair. We're doing pretty well, and Sanger's doing a little bit less well. If you look at heterozygous errors, well, we're doing the best. Sanger's pretty close, and the Broad's clearly worse. And if you look at non-reference errors, Sanger's doing better, we're doing okay, and the Broad's the worst. And so obviously we looked at this and said, clearly the Michigan results are the best. That's what we should choose. But it was hard to get to a consensus across the entire project that was the right interpretation. And so after kind of many hung discussions, one of the bureaucrats in the project, we have some funders that join our calls and said, why don't you guys come up with a consensus? Why don't you have 3 answers, why don't you take a vote? If 2 of them agree, then you'll pick that one at each position. And, and to me, this seemed like a terrible solution, right? You have 2 bad methods, but they outvote the better method or something like that, right? But, because they were paying the bills, we said, we'll try it. And so that's the bottom row here. And it turns out that actually doing this kind of consensus approach greatly reduces the error rate. What's happening here, actually all the methods are not using all the information that's in the data because it's too complex. And so they each made different approximations and decisions. When you combine them, you do much better. It's the same thing when you look at things like predicting what a Netflix user wants to watch next. It turns out that you can use many sophisticated statistical models, make a prediction, and the best possible prediction is by running a few different models and letting them vote among themselves. So kind of cool, but it also says, hey, you actually don't want to just spend those 400 CPU months. You want to spend them a couple of times or something. OK, things that you get to see when you look at the data in the end. Here's a summary of genetic variation around transcripts. And so you see, for example, that there's a clear depletion when you are right on top of any gene, and that depletion actually extends for about 0.1 centimorgans, about 100 kilobases. So it's kind of nice to see some of these things that you'd predict, natural selection is occurring at the gene level, but actually quantifying how far from the gene it extends and so on. As the project has progressed, this was our pilot paper, we've added more individuals and discovered more variants. We're now at about 40 million. And one nice thing about these methods that borrow information across individuals, when you look at the error rates, we're also getting much more accurate at how we call each individual genotype. The most recent release, about a month ago, we actually integrated all different types of variation. We were analyzing the large deletions, the single-site SNPs, and the indels into a set of chromosomes for each individual. So, and this data is used in many different kinds of disease studies. You can use it in resequencing experiments to screen out variants that are likely not to be disease-causing. If you are interested in fine mapping a region or studying it in detail, you can look up all the variation that's been seen in 1,000 individuals, design an array to inexpensively query it in your own samples. You could actually try and connect the haplotypes we see here to haplotypes you've generated with lower resolution and fill in data by imputation in your own studies. So here's a different sequencing project, this one with disease samples. So here, two key collaborators are Francesco Cuca and David Schlesinger, so their names are there. And so what are we doing here? We have a population of about 6,000 individuals recruited from 4 towns in an isolated valley in Sardinia, which is an island off the coast of Italy. The total population of the valley is about 10,000. So we have about 60% of the people enrolled in the study. They come in for a medical exam every 3 years, and we phenotype them in detail. So we have a series of quantitative traits related to aging, ranging from things like blood pressure to cognition and so on. And we originally set out to sequence 1,000 individuals at least 2x coverage, which is quite low, but at the time was what we could do with our budget. And without having spent out our budget, we now have 1,700 individuals sequenced at almost 4x. So we have 4 times more data than planned already, and we're not done yet. So when we wrote the project, we assumed sequencing costs are going to go down by a factor of 2 every year. And the reviewers said, maybe that's a little bit aggressive, But in fact, they've gone down by a factor of 4, and so we have already quite a bit more data. One nice thing, they have a very nice team of hardworking people there in Sardinia, and I like to say that we print out the sequences in those pieces of paper, and then we manually align them and so on. So it's not quite true. Okay, so designing the study, how do you set up the details? How do you figure out who to sequence? So because we have pretty much the entire not in the entire, a large part of the population in this valley, we have lots of relatives. We can find many families that look like this, and we can make decisions about who to sequence. It turns out that if I sequence the 3 individuals at the top of that pedigree, the ones that are shaded, I actually am able to deduce the genomes of their relatives because it's effectively the same chromosomes that are moving through the family. And I'm going to also be able to deduce half of the genome for the bottom generation. And for in a relatively effective manner, I can add a few more people, sequence them, and be able to deduce the sequence for the entire family, getting me a further cost savings. So this is just to see, how sequencing is progressing. This has mostly been the work of a postdoc working with the sequencing core here in Michigan. March 2010, when we started, we were getting about 5 gigabases of data for a fixed amount of cost, a fixed amount of effort. Now for the same amount of cost and effort, we're about 35 gigs. So when you see these things saying the throughput is increasing 4-fold each year, it's really true, and it's kind of nice to watch. And what happens when you look at a particular trait and find out what you learn? So here's LDL cholesterol. So LDL cholesterol is an interesting trait because it's a very strong risk factor for heart disease. When we do a scan, here in red you are the results of our previous scan based on a standard genotyping array, and in gray in the background are the results from a sequencing study. The sequencing study has a lot more markers. We're looking at 20 million markers instead of 2 million. Most of the results are concordant. So you see a big peak here at ApoE, another peak here at LDLR, PCSK9, SORT1, and ApoB are all genes have been implicated for quite a while in LDL cholesterol metabolism in humans. And quite a while here means, in some cases just 2 or 3 years. But it turns out there's also a very strong peak here, which is a stop codon in the beta-globin gene that had been completely missed. It had been missed because this variant is unique to Sardinia, so it's not in these common variant arrays, and it's relatively rare. If you go and examine it in more detail, it turns out that this particular stop codon is the biggest determinant of LDL cholesterol levels in this population. It's associated with a large increase in turnover of red blood cells, and making these new red blood cells ends up consuming your cholesterol. So you have much lower cholesterol if you have this variant. And in heterozygotes, it appears to reduce your risk of heart disease and things like that. So it's kind of a nice story that we would have missed if we just used the standard technology. So it's kind of nice to see it coming back. Okay, so what I wanted to give you is a flavor that sequencing lets us do things we couldn't do before. Getting to sufficient sample sizes is still a challenge. We'd really like to sequence hundreds of thousands of individuals. Here we're talking about studies with low thousands. So we need to think very carefully about good designs, interesting samples. And I try to give you a flavor of some of the challenges in analyzing the data, and it's at least as tough as generating it. And I think when people talk about, well, when the costs drop enough, you're going to have your genome sequenced for a few $100s or maybe less. And on the current trajectory, the analysis might cost more than that. Okay, so these are the people in my group here in Michigan, key people here are Hayun, who does a lot of our sequence processing pipelines, Carlo, who did all the analysis for the Sardinia data, and Fabio, who actually generated most of it in the lab. So, all right, so thanks.
Any questions for Dr. Abecasis?
Yep. The 1,000 genotypes that you get out there, the number of whole genomes you sequence, are you starting to stop seeing new variants, or are you still on the trajectory of having what you identify as new?
So you see new variants in every genome. So the largest data set we have is an amalgamation of different sequencing projects where we have 12,000 individuals exome sequenced, and there more than half of the variants are seen in a single individual. And actually there's interesting reasons to try and get them all, because say variants like stop codons, things that disable a gene, are extremely rare. But you could imagine that it would be great if we could have a catalog of where we have one person with a stop codon for every gene, because then if we want to know about the function of the gene, we could call that person back and do some tests on them. Well, to get to that, we will probably have to sequence well over 100,000 people deeply. So there's very interesting experiments that you can do if you can reach that kind of scale, and there's many variants that are rare and interesting. So, yeah.
What tool were you using?
Okay, what part of the analysis? Sorry.
Well, I'm wondering specifically like the haplotype analysis.
Okay, so for doing the haplotype sharing-based calling, we use a tool that we developed here called MAC, and it basically, uses hidden Markov models to search for shared haplotypes among large sets of individuals. The upstream steps are using things like BWA and SAM tools and GATK, and a combination of them most of the time. All right, so I think I should.
Any other questions? Thank you. That was fantastic. Look at this. This factoid about the Nature paper is like stunning. Let's all, our hats off to you. This is absolutely great. And as far as the investments, we're making significant investments to enhance the cyber infrastructure at enterprise level to allow for us to grow a DNA biolibrary to 100,000 specimens, annotated with health record, safe and secure and private to anticipate the HIPAA ruling that's probably going to come down the pipe, and we're really proud to be working with you. Really cool.
I mean, you can only do this in a place like Michigan. It takes lots of good people.
I was telling Jim that yesterday in my meeting with Ted Hans and Dr. DeWitt, who's our CIO on the health system side, that it wasn't the 40 million for the sequencing, but it was at least 5 or 10 million to get some of the infrastructure in place to help you. And we really are working hard to get this working in the institution. Okay, we've got our final speaker up here.
Well, I'd like to thank the organizers for giving me the opportunity to share my excitement about the power of genomics and bioinformatics in modern biology. And I want to tell you a little bit about research of my lab, which is going to be very different from what you just heard. So I will start with a brief background about our research questions, and I will tell you about one specific genomic project that's going on in the lab right now, and I will tell you a little bit more about the tools we use and bioinformatic approaches we try to apply in the lab. I will tell you very briefly about findings of our research so far, and I want to finish with a take-home message about bioinformatics here at U of M from a biologist perspective. Well, I'm assuming that everyone knows what the genome is, and that the human genome has been sequenced over 10 years ago. And it wasn't the first genome to be sequenced. There were bacterial genomes and the yeast genome sequenced earlier. And right now we have probably thousands of various organisms with sequenced genomes. So everyone thought, wow, great. We'll know the sequence of the human genome. We'll know everything about human. And 10 years later, that's a pretty big disappointment. So the sequence of the genome is huge, is cryptic, and we have no clue what it means. And it's a really big challenge to really understand what the genome is doing, how the genome is regulated, and how life translates information from the sequence of nucleotides in DNA to us or to other organisms. So the big goal of the big part of biology, including my lab, is to understand how the genomes work. And probably one of the reasons why I was asked to be here is to show you that biology is not only human, it's not only biomedical research, and that's true. So I'm doing my research on plants, and in general, model systems and plants specifically are really convenient. There is a lot of questions which are very hard to ask in the human system for practical reasons, for ethical reasons, and in the model systems we can ask them really easily. So plants, even though many of you who are in the biomedical field are not really so excited about plants, plants are actually really good models for biomedical science, because plants are eukaryotic and they are multicellular. So plants share pretty much majority of the basic mechanisms with human and with vertebrates. At the same time, they are much easier to work with than animals. A lot of practical and ethical and other concerns are really not a factor. Although plants have their own specificity, there has been specific fields where basic research has been done in plants much more effectively than in other systems. At the same time, plants are really exciting on their own. They are the major harvesters of energy, so they are critical for this world. And they do a lot of really fascinating stuff, and I shouldn't even start talking about this. So the specific question we are asking about the genomes is how non-coding RNA regulates activity of the genomes. So let me introduce you real quick to the non-coding RNA. So the very basic fact about life, and something I usually teach in one of my first genetics lectures, the information in living organisms flows from the genome, which is DNA, to proteins, and proteins perform the actual function. So they build our bodies, they are enzymes, and they actually do stuff. But there is an intermediate, and this intermediate is called RNA. It's also a nucleic acid. It's very similar to DNA. It's only one atom being different between nucleotides in DNA and RNA. And there are many reasons why this intermediate is needed. And for years, people thought that RNA is just an intermediate. However, it turned out that there is a whole lot of RNAs which are not intermediates. They are doing something else. And some of them have been known for years now. And the research of the genomic era made people realize that actually the majority of the genome may be encoding RNA, which are not encoding proteins. So these are the non-coding RNAs. And trying to understand what they are doing is the specific goal of my lab. So let me shift to even more specifics and tell you about the specific project in the lab. And this is the project where we successfully used the genomic approach, and we learned a lot of stuff in the process of working on this project. So we want to understand what a specific class of non-coding RNA is doing on the genome-wide scale. And it is important because there has been a lot of research on various regulatory proteins and RNAs which would study only specific fragments of the genome. And people really love to generalize. Okay, we found these 2 genes which are regulated by this non-coding RNA, so it means that this non-coding RNA is doing whatever it's doing on the whole genome. But it wasn't really supported by any data. So that's why the genomic approach is really important to allow us to make general conclusions about functions of various factors. So we try to study a class of non-coding RNA by studying a protein, which we knew already before, that binds to this non-coding RNA. And we knew that this non-coding RNA is guiding the protein to DNA. And this protein is called Argonaute. It is involved in controlling activity of various areas of the genome. So what we wanted to do, we wanted to find where this Argonaute protein binds in the genome, and we wanted to use this information to understand what is non-coding RNA doing, and hopefully also understand how exactly it is working. And to achieve this goal, we performed a ChIP-seq experiment, and there is a lot of words on this slide, and you can see the very top. But it's in principle a very simple method. What we did, so what we are doing, we are grinding plants in liquid nitrogen, extracting proteins, and then we are purifying our protein of interest. In this case, it is the Argonaute protein. And when we purify the protein, associated DNA is co-purified. So if the protein binds to a specific region of DNA, if we purify the protein, we'll also purify DNA of this specific sequence. So we can get this DNA and sequence it using next-generation sequencing, which allows us to get, nowadays, 10 million of reads per every experiment. And having this data, we have to analyze it to understand what is the function of the protein. So let me tell you real quick about the way we analyze the data, and then I will show you a small sample, and then I will tell you a little bit more about the tools we actually use to analyze the data. So we always have to start with mapping the sequencing reads to the genome. And once we map them to the genome, we can look at the specific fragments of the DNA. And if we see a lot of reads mapping to this region, more than in our control, we can say that our Argonaute protein binds to this specific part of the genome. The next step is to visualize it somehow. And I will show you an example of visualization in a moment. We don't have to find significant effects, so distinguish what is random from what is a biologically significant binding of the protein. And then we have to do something which I called creative analysis or a downstream analysis. So basically start with this enormous data set and then do some magic and end up with understanding. And I will tell you a little bit more about this last part in a moment. So a really important step is to visualize the data. And this is an example how it looks. And you can see that this is a fragment of the Arabidopsis chromosome. And here on top are genes. This is one gene. This is the second gene. And here, well, this part up here comes from different sets of experiments. So let's just forget about this top part right now. And these three lanes come from our Argonaute 4 ChIP-seq experiment. So, and this is our experimental lane, and this is a negative control. So you can see over here that there are some sequencing reads aligning, but they are in both in our experiment and in the control. So there is no significant binding here. But in this region here, there is a lot of reads aligning in our experimental line, but much less in the negative control. So we can say that this part of the genome is associated with our Argonaute protein. Okay, so let me tell you real quick about the tools we use. And so the first step, mapping to the genome, it's pretty routine. And in our application, we just have a simple Linux workstation, and because Arabidopsis genome is kind of smaller than the human, not much smaller, but it is smaller, we can just map it. It takes a few hours to map a pretty large experiment to the genome. And the software is pretty standard, so that is not a big challenge. Then to visualize, what we need is a web server. And it's actually not so easy to get a web server up and running for many reasons. But most visualization software operates as web servers for, well, for us to be able to use it first, but then after the data are published, to make it available to the community that other people can browse through our data and use it. The next step, finding significant effects, or we call it that it's peak calling, also uses just a simple Linux workstation. However, the software is usually kind of specialized because there's a ton of various peak calling methods, and our experiments are usually not something really standard. So in practice, what we have to do is to dig through the literature, find like 10 or 15 various algorithms, try them all with various parameters, design smart controls, and using those controls, decide which approach works best. And the final step, trying to convert this data set into understanding, this is actually the core, the main challenge and the most exciting part. And it doesn't really take much of computing power. We still use our Linux workstation, although I could easily do it all on my laptop, which I use for this presentation. And there is a bunch of specialized software, and we also have to write scripts and small pieces of software on our own. And the general approach we take is we look at the visualized data, look through the genome, and try to make observations. And let me just give you an example. For instance, you may see that this binding region is aligned pretty well with start of a gene. And that's something we actually observed over several genes. So we hypothesize, okay, Argonaute 4 binding is correlated with starting points of genes. And now we have to move on and test it on a genome-wide scale. So take all genes from the whole genome and look if there is Argonaute 4 binding on all of them. And to do it, if we are lucky, we find a specialized piece of software online. If we are unlucky, we don't find it, we have to write it, which is usually not really difficult, even for a dumb biologist. Well, try to get our answer and try to design a control to make sure that our answer is not wrong and our software has no bugs, and everything fits into a story. Let me tell you real quick about our conclusions, which I'm really super excited about, and I'm afraid that I wasn't able to give you sufficient background to make you the same excited, but hopefully at least you'll understand at least a little bit of my excitement. So we were able to find where non-coding RNA is guiding this Argonaute protein, and it turned out to be a very different part of the genome than what was expected. And those of you who heard about Argonautes know that this protein is involved in gene silencing. So the expectation was to see it on the silent part of the genomes, and there are huge areas of the genomes which are permanently turned off. And we're expecting to see this Argonaute protein on those huge inactive areas of the genomes. But we don't see it there. We see it exactly in places like here, in gene promoters. And promoters are very important pieces of genes. These are decision makers. These are fragments of DNA which decide when and where a specific gene is turned on and turned off. And to maybe make the point even stronger, the pattern of gene expression is in many cases much more important than the protein which is encoded. And for instance, comparing human to monkeys, the protein coding part of the genome is extremely similar. And many people think that it's the pattern of gene expression, so when and where the genes are expressed, that actually makes the difference between human and monkey. And so that's why we are very excited to see this Argonaute 4 protein and non-coding RNA function specific on gene promoters. And well, our hypothesis is that this non-coding RNA we are studying is specifically targeting promoters of a pretty large number of genes. This is like in the 500 to 1,000 range. So the large number of genes and is some kind of regulatory mechanism which affects transcription of those genes. And our very preliminary data suggests that it may be actually involved in stress response. That the genes which are targeted by Argonaute 4 and non-coding RNA seem to be, most of them seem to be regulated by stress, and we think that this Argonaute protein may be actually involved in stress response. Okay, so let me finish with a take-home message about our experience at this university, and because I hope that in this room there may be some decision makers who may make our life easier for the future. It's really remarkable that nowadays computers became standard research tools, like a pipetter, like a centrifuge. We also have a computer in the lab, and all we really need, except winning with our with biological dampness and our difficulties in writing software and understanding bioinformatics. The other thing we need is flexibility. So we need to be able to try as many tools as possible, and we need to be able to follow evolving bioinformatic tools. And it takes installing new software 3 times a day. It takes doing, well, whatever idea we have now, we want it done 2 hours from now. And unfortunately, this is probably very specific from unit to unit, but this university has a lot of regulations about computers. And it happened to us several times that we wanted to do something, and we heard, no, you can't do it. This flexibility is really important to use computers for research efficiently, especially for research like ours, which takes a lot of trial and error and a lot of new tools being tested and developed all the time. And I think it's important for the administration of the university to understand that every limitation, and so every time someone takes a piece of this flexibility away, research, it interferes with research. And every time a piece of this flexibility is taken away, competitiveness of our university is also reduced. So I really hope that the university is going to invest in cyber infrastructure not only by building new supercomputers, but also by helping us being able to do whatever we need to do. All right, so let me finish with thanking members of my lab, especially Jordan, who did most of the work I was showing today, and other present and past members of my lab, and our great collaborators, Brian Gregory and Chi Zhang at UPenn. And thank you for your attention, and if there are any questions, I'll be happy to answer.
Great, thanks. Any questions? I did see some information technology professionals nodding their head. I guess one thing I would like to say is that sometimes the administration stands in the middle between the government and the faculty, and we are being barraged by compliance and regulatory issues right now, and we're doing everything in our power to try to understand those issues. We limit the amount of red tape, and at the same time increasing the flexibility. That's our commitment. Question? The recording is going. Oh, okay, good. See, I missed the microphone.
So, if you could identify maybe a key bottleneck for you, what flexibility don't you have that you would like to have, and is that something that maybe as a community we can help you achieve?
Actually, the biggest bottleneck has been resolved, and the breakthrough was when we got our own workstation and we convinced our departmental IT to get us the root password. And at that day, everything, well, almost everything has been solved. But they made a big exception for us by giving us the password. It's something they generally don't allow. Okay.
You know, I think we have an opportunity to learn from you. You know, there's a big biomedical focus to a lot of the computing that's being discussed here. And you're coming from campus side and biological focus. And so, you know, from your perspective, you know, what I'd like you to do is, you know, kind of, you know, give us some feedback on this difference and, you know, what we can do to maybe bridge the gap between the biology and the campus side and the health.
Yeah, so actually, that is a very good point, and I think that our university is kind of strong about it. And I have had experience from a different institution where I was a postdoc, and when I was emailing the sequencing core, if there was the word "planned" anywhere in the email would never get a response. And here I am getting responses from the sequencing core, even if I write "planned," even if it's in the subject of the email. Well, I think it's important. It's really important for the biology part to really be on top and do really a breakthrough research, do stuff that's interesting enough to interest the biomedical folks as well.